I want to welcome everyone here to the Wilson Center. My name is Todd Kukin. I'm a research associate here with the Synthetic Biology Project. I also have worked on our project on emerging nanotechnologies. So I want to thank everyone for coming. I want to go around the room quickly just to get sort of your name and sort of who you're with, where you're from. We don't need any sort of long bios, and I'll do that. And then after that, I'll sort of explain how we're going to run things today. So why don't we start? Joe, can you grab one of those wireless mics? And we're live webcasting this, which is why we need the microphones. So why don't we start over here, and we'll go around the room real quick.
I'm Kathy Dolan. I'm with Food and Water Watch.
I'm Ross White from the Hastings Center.
I'm Janice Meyer from Sierra Club.
James Sang, recently retired from IBM Research, and I'm a physicist.
Brian Greenberg, Interaction.
Karen Hansen-Kuhn, Institute for Agriculture and Trade Policy.
Joe Freeman, Senior Scholar, Woodrow Wilson Center.
Amanda Wilson, Interpress Service.
Marcy Wright, Health and Human Services.
Jerry Ostheimer, USDA Foreign Agricultural Service.
Jeanette Cruz, private citizen who thought it looked interesting.
Colleen Cordes with the Loca Institute.
Jesse Coleman with Greenpeace US.
Jenna Jaden, USDA.
Michal Rosenauer, Friends of the Earth.
Julia Birch, EPA.
Hello, I'm Daryl Ditz, the Center for International Environmental Law.
Bashkut Tunjak with the Center for International Environmental Law as well.
Larry Lanes with the U.S. Department of Energy.
Edwin Bajak, no affiliation.
Khaled Bori, FDA.
Leah Middlemass with the Australian Embassy.
Dylan Castine, Environmental Working Group.
Alex Rindler, Environmental Working Group.
Janya Dana, Woodrow Wilson Center STIP Program.
Sarah Carter, I'm the Policy Analyst at the J. Craig Venter Institute.
Johnny Dell, a retired engineer.
David Barton, retired. All right, thank you very much. So the main reason for that we decided to have this meeting and workshop today was to sort of bring the larger NGO community into the discussion of synthetic biology. Part of what we do at the Synthetic Biology Project is to incorporate all of the various players as this technology is developing. And we've been working with Eric and JD here for quite a while, I would say. JD a little longer with our nano project. So we asked them to come and give the perspective of their particular organizations and a little bit of the larger NGO community overall's concerns and interest in synthetic biology. So how we're going to sort of operate this today is JD Hansen from ICTA is going to speak first on synthetic biology, life redesigned. Then I believe Eric Hoffman from Friends of the Earth is going to focus a little more on biofuels and the environment and public health issues. And then Kathy Jo Wetter from ETC Group is going to talk a little more at the economical implications and probably some other issues as well. And then we're going to finish up with David Rajewski, who is the director of the Synthetic Biology Project here, who's going to discuss some recent focus groups that we just completed last month, where we brought together some people, locked them in a room, and had them discuss their feelings on synthetic biology. So with that, I am going to turn it over to J.D. Hansen.
Well, good afternoon, and thanks for coming out. My task is really to talk about what many of the whats are that are being called synthetic biology. When we first designed this, we were designing it mostly for the environmental community that may not have worked much on synthetic biology so far. So bear with me, those of you that work even doing synthetic biology, if I'm pretty basic going through this. The International Center for Technology Assessment was formed I believe about 17 years ago. Our name is not an accident. We were formed right after the Office of Technology Assessment was shut down by the Congress, and our founders thought it would be good to have a group that still tried to think about new technologies and what they might mean and how the society ought to deal with them. Unlike the old Office of Technology Assessment, we don't take government money. They were funded by the Congress, and we don't take money from corporations. Our money comes from individual donations and foundations. I get into this after having done work in fisheries, done work in environmental policy, and then a considerable stint doing biotechnology and bioethics, working with the United Methodist Church. Okay. Synthetic Biology 101. Well, the old genetic engineering was basically moving one or two genes from one organism into another organism. Synthetic biology, and I'm borrowing the term from my friends at the ETC group, might be called extreme genetic engineering. One of the ways of doing synthetic biology is to use a computer to print out the chemicals that make up DNA and RNA, to make a sequence for a gene or a genome, or a number of the researchers hope a whole organism. This is a big difference between the old genetic engineering and this new synthetic biology engineering. Again, the old genetic engineering was just a few base pair or pieces of DNA or RNA. The new synthetic biology hopes to copy large stretches of DNA, over 1 million base pairs for even the first small genome that was copied. And the temptation, if you will, about synthetic biology is really powerful. And the engineers that are working in this field want to harness that power, because right now even the simplest organism is more complex than anything we humans have yet invented. And my engineering friends might argue about that when you look at the highway system, but anyway, it is a field that is moving fast. Within the last year, Dr. Isaacs and Dr. Church have developed a new method for, they're at Harvard Medical School, for making hundreds of changes in a genome simultaneously. I'm skipping ahead, though. Let me show the antique. This is a DNA synthesis machine now in the Smithsonian. That's where our antique machines go. DNA synthesizers automatically construct short sequences of DNA, which can then be assembled into longer molecules, including single genes or complete genomes. This technology became commercially available in the early 1980s. In 2002, researchers used a machine similar to the one above, and they took a 1981 description of the poliovirus and used it to synthesize the poliovirus. Since then, other teams have synthesized the 1918 Spanish influenza, smallpox, and many other viruses. This demonstrates that DNA and RNA synthesizing machines can write more than just strands of DNA, but also whole genes and maybe whole organisms. Now, such machines, while they were powerful to write, machines like this one were powerful enough to write the poliovirus and other viruses, they're considered obsolete. If you don't want to go see one at the Smithsonian, you can look on eBay. I last found one being advertised for $1,000. Nonetheless, synthetic biology is a major change for how biotechnology works. It means that you can use biotechnology to make larger proteins. You can go through many more engineering steps in traditional biology. And so synthetic biology is now using small molecules, proteins, RNA, DNA, metal ions, changes in temperature, and pH to develop programmable input and output tools. The bottom line for trying to do all of this on the researcher side is they hope this will allow faster developments of cellular therapies, faster developments in agriculture biotechnology, biosecurity tools, and diagnostic devices. A few years back, some computer engineers at MIT decided that they really wanted to figure out how to standardize genetic engineering in a way that made more sense to them as engineers. And so they developed, they said when you're making a ordinary engineering project, you know which parts you need, you can order them. Why don't we figure out how we can make biological parts standardized so that we can order them and make new organisms? The language, and you can't quite see it there, that they use for these little parts of organisms, promoters, protein domains, protein coding sequences, translational units, terminators, sounds an awful lot more like terms that engineers are used to using than the biology that I studied years back. Part of that's because biology's changed. Part of that's because the intersection between computing and biology has moved rather fast. One of the names used for this approach is BioBricks. There's actually a foundation based out in California, the BioBricks Foundation, where you can order these different parts through. And there's a group of students now that are ordering these parts and contributing these parts called the International Genetic Engineered Machine iGEM Competitions. And this is last year's gathering. Todd is going to be a judge for this year's team that is meeting in Amsterdam, right?
Well, it's regional now because it got so big.
Okay, it got so big.
Back at MIT in November.
All right. Well, I refer to this as your high school science fair on steroids. This group of students are using the so-called BioBricks approach to synthetic biology, and last year, at least at iGEM 2010, they awarded prizes in 8 categories, from food to software development, and some of the projects are quite interesting. The one that won the Best Environment project was a team from Beijing, China, and they developed a method to engineer bacteria into heavy metal decontamination kits, and they basically were looking at how to detect and deactivate mercury in the environment. I was happy to learn just yesterday that Todd had been on the biosafety branch of this team, and that they one of my criticisms was I didn't think their biosafety protocols were very good, and Todd, if he wants to, in questions, can talk more about how that's worked. But it does seem to me from talking to Todd that it's working better than it did before. This is a picture from the J. Craig Venter Institute website of the mycoplasma. This is Mycoplasma mycotes. It's a bacterial goat pathogen that became the first entire genome of a living organism to be copied using DNA synthesizers. In an article published in May 2010, Dr. Daniel Gibson and a team of 23 other researchers at the Institute outlined the steps that they used to synthesize a 1.08 million base pair genome. This genome was then booted up into a cell to create the first cell completely controlled by a synthetic genome. They weren't able to get the genome to boot up first without being inserted into another cell, but that's their next challenge. The boot up language is from the researchers, not from me. Again, a way that computer language has entered into this jargon. I'm actually going in what I think is the order of development in the field. Depending on what you want to call it, this was one of the pejorative ways of describing this genome-wide codon replacement was genome hacking. But regardless, this is a team of MIT, Harvard, Yale, Stanford researchers, and they demonstrated that they could replace wholesale all of the tag stop codons in the E. coli genome. In their report, they say introducing genome-wide changes dynamically in a living cell permits engineering the cell's native biological contexts. In contrast to in vitro genome synthesis and transplantation methods that introduce discrete and abrupt changes in a single genome, we can now treat the genome, the chromosome, as editable and an evolvable template. We were talking a little bit beforehand on some of the interesting intersection between religion and science these days, and I think one of the challenges is what kind of language is used. The Ventnor paper talked about creating a genome, and I asked one of the researchers, why did you choose that? And their response was, well, I wrote build, but the boss crossed it out and wrote in create. For the scientist here, I would say be careful about loaded phrases like create and evolve, as we want to talk more with the religious community about some of these things. Those of you that know me know that I like puns. This is actually a pun that Johns Hopkins publicist used to talk about Jeff Boak's laboratory's work on yeast chromosomes. a call to arms for synthetic biology, ? Scientists at Johns Hopkins announced just 2 weeks ago that they have replaced all of the DNA in an arm of a yeast chromosome with computer-designed, synthetically produced DNA that is structurally different from its original DNA to produce a still functioning yeast cell. Now, yeast chromosomes are often depicted as bow tie shaped, with each chromosome bearing 2 arms that are presented similarly to the 2 parts of a bow tie. What this study confirms is that large pieces of DNA can be synthesized and inserted into a chromosome, and they serve to validate the research team's way of designing synthetic chromosomes. The researchers report a method for changing the structure of synthetic DNA, a process they call scrambling, which can be applied to other organisms in addition to yeast. One of the reasons that they, so far, this isn't the whole yeast chromosome. This isn't the whole yeast genome. It's about 1% of the genetic material in the yeast. But they want to synthesize all 16 yeast chromosomes in order to give the organism their desired traits. Yeast, I would note, is used in many processes, and including, we're talking basically about what makes bread, what makes beer, and what's being used to make vaccines, and what some folks hope will be used to make biofuels. Therefore, Boak's lab thinks that giving yeast the ability to more efficiently confer desired traits on this organism may lead to the production of new vaccines and more efficient biofuels. I guess to get funding, we're past the point that we do things just because they're fun to do. So everything is done because it's going to cure you. Everything is done because it's going to get us weaned off of Arab oil. Again, watch the language. One of the bioethicists at Harvard, not Harvard, at Duke, Stanley Hirawas, says people used to want to know why God allows suffering. Now they don't ask that question anymore. They want to know why in the world science hasn't cured cancer yet. So I think while the research is really interesting, I would again urge the scientists listening to be careful about the promises there. Sometimes the public believes those promises and wonders what's taking you so long. And as we're dealing with all of the debates on funding, none of this in some ways is really, really synthetic, because we're all playing with existing life forms. What if we could make life that never existed before? And there's actually a field of synthetic biology that's trying to do that. Some of them call what their work is protocells. They're trying to build life from the bottom up. They're trying to build organisms that reproduce, grab energy, get rid of waste, but aren't based on DNA. Dr. Mark Badeau, a philosopher turned synthetic biologist, believes that one of their teams working on bottom-up synthetic biology will succeed in the next 10 years to make a life form that's never existed in the universe before. Now, that's a pretty radically different idea, and it's not being done yet, but I wanted to mention it because there's some serious discussion. Now, what is being done? There are folks working on engineering new drugs, new stem cell lines, folks working to engineer viruses, engineering with pieces of microorganisms. The biomachine competitions is a good example of this. There's actually a field, do-it-yourself biology, that are using these pieces to create in their home labs different creations. I'm using that word create again. There's work being done to make biopharmaceuticals, and there's work being done to make biofuels. There's efforts being made to engineer whole organisms. I mentioned the Venture project and Jeff Book's projects. Algae, bacteria. Some of the scientists talk about plants, animals, even humans being engineered with these techniques. While the U.S. President's Commission on Bioethical Issues thinks that existing regulations are adequate for synthetic biology, I think the three of us up here would disagree. And I would say, in my opinion, that right now only new drugs created from synthetic biology are really being regulated in any significant way. And that is unfortunately mostly through the secret FDA drug review process, and even the recombinant DNA advisory committee of the National Institutes of Health agreed, at the request mostly of drug companies, that they won't review projects involving fewer than 100 base pair. I'll move on. I'm running behind time, right? Yeah. Let's look real quickly at a couple things that are being done. The Amyris company has the distinction of having the first products that they claim come from synthetic biology, and they do come from yeast that are engineered with some synthetic sequences of DNA. They first developed their technology with a grant from the Gates Foundation, and they were able to develop a platform technology that used yeast first to produce artemisinic acid, a precursor of artemisinin, one of the anti-malarial therapeutic drugs. And they licensed their technology to Sanofi Adventus for the commercialization of the drugs. Now, this might not have been my first choice, as awful as malaria is, in that you had a problem that was really a supply problem, and that the Chinese wormwood prices go up one year and they go down the next year, and the peasants decide or not whether they're going to grow Chinese wormwood, which is used to make artemisinin. We know how to deal with supply problems, and we don't actually have to engineer yeast to do it. We do it with dairy pricing all the time, and we give good signals to farmers that they produce things year in and year out. So we may have a new supply of a drug, but it will be from a company based in Europe and not from farmers in Africa or China. and sadly, because artemisinin is rather widely used, malaria is developing resistance to it. So we're going to be looking for a new drug pretty soon, anyway. I did mention that they were going to that they're the first company to actually make money on their products. Oil on your hand, that oil is mostly farnesene. Oil on your forehead. Farnesene as a good oil. You can maybe eventually use it for biofuels, but what the company's found is they can get a cosmetic company to buy their genetically engineered farnesene. They basically engineered the yeast to make this oil. The yeast really like to make ethanol, but they've got them to make a little bit less ethanol and a lot more farnesene. And they now have a market for it, and I don't know which company it is, but it's their first sale. And you're going to be talking about all the feedstocks that they're doing in Brazil, right? Okay. Yeah. This is synthetic genomics, Craig Venter's for-profit arm. They have been joining with a couple of oil companies, among them BP and ExxonMobil. In 2007, they announced that BP had made an equity investment in the company to sequence the genomes of naturally occurring microbial communities that live in oil. The goal is to make organisms that produce biofuels, petrochemical substitutes, and vaccines. They've also received a promise of $600 million from Exxon for its biofuels work with blue-green algae. When I was out at their labs talking with their staff, I said, well, if you really were to be successful, how big, what size ponds would you need to get? And they said, well, we've done some back-of-the-envelope calculations, and we think about 70 square miles. Now, even if that's just back-of-the-envelope calculations, 70 square miles is a pretty big pond. That's the size of Washington, D.C. I said, where are you going to put that? They said, well, we don't think we'll be able to do those, that scale of ponds in the U.S., so we're talking to some other countries. Well, some of you may know the work of Lux Research. They do a lot of profiling of what kind of investments are going into different new technologies. They estimate that last year $500 million went into synthetic biology investments alone. That's not a small amount of investment. This overall chart actually looks at all of the investments in some of the more cutting-edge biofuels, and synthetic biology gets the lion's share of the. Now you can also see that the investment has gone down, as have a lot of other investments in this climate. But still, synthetic biology is the investment that really has gone up and maintained its share over this time. Now, I would point out that this chart is private equity funds. This is not government. This does not include government funding. The National Science Foundation, Department of Energy, and the Defense Department are major funders of this research as well. our office is working on a new report on government investment in synthetic biology that should be published by the end of the year. And we'll get back to you on all that. And I was going to talk about DARPA, but any of you live in Arlington, Virginia? Any of you ever wonder what that building is over by the Starbucks at Virginia Square? That's DARPA's Arlington office. That's why there's always these, black security vehicles around there. DARPA is the really geeky part of the U.S. military. It's the research arm that likes to remind us that it invented the internet, not Al Gore. But it now wants to use computers to redesign life through its Living Foundries program, and that's I'll save for another day. Thank you for your patience.
All right, thanks, JD. We're gonna hold questions to the end. We can have a larger discussion about synthetic biology in general, what's been said today. So I'll turn it over to Eric Hoffman from Friends of the Earth.
Great, thanks, Todd. Again, I'm Eric Hoffman with Friends of the Earth. We've been following biotechnology for a number of decades now, first in genetically engineered crops and now in synthetic biology. I'll also say that we, too, don't take corporate or government funding as well. So thanks to JD for that really good background. I talk much quicker than JD, so hopefully we'll catch up on time. But today I'm going to talk about the mostly environmental, and I'll touch a little bit on the public health risks of synthetic biology. Just to start, I think it's important to note that many of these risks are unpredictable. Some of them are pretty similar to things we've seen with risks from genetically engineered crops, such as genetic contamination, which has already caused some serious problems, both economic and environmental harms to farmers, which the Supreme Court agrees when they ruled in the Center for Food Safety case in about genetically engineered alfalfa. But some of these risks are new, or they will become more and more novel as the field of synthetic biology grows. As JD mentioned, we're dealing with more novel genes, novel traits, and novel organisms that have never existed before in nature. As the technology develops, we're seeing that synthetic biologists can create more and more organisms at one time that are all different. So you could eventually have thousands of different new organisms being created in one experiment. And the ability to assess risk when you're dealing with thousands of novel organisms that are each different, it's a very different ballgame than just assessing the risks from one genetically engineered corn crop, for example. And I'll also argue that the risk assessments and regulations around this technology is lagging behind, and the technology is really jumping ahead quite quickly as the regulators and policymakers are slow to catch up. So environmental harms will mostly come from different forms of environmental release of synthetic organisms. These organisms could be released unintentionally from a lab, so a failure of containment maybe, or a natural disaster hitting a lab, or it can be done intentionally. Some hope to use synthetic organisms for such things as bioremediation, or even as new forms of pesticides or herbicides in agriculture, so actually spraying these organisms out into the environment. And I'll also argue that commercial use of synthetic biology is also the same thing as the environmental release of synthetic organisms. A lot of the work being done, as JD's mentioned, is on bugs such as algae, yeast, E. coli, and viruses. All of these things can easily become airborne and escape confinement, and they escape confinement in labs all the time. As Jay Kiesling, the founder of Amyris, the company that JD talked about briefly, said that their process of using synthetic yeast to produce cosmetics and biofuels, he says it's, quote, very similar to producing beer. And if anyone knows anything about the beer industry, if you're a home brewer or whatever, contamination is a serious problem. You have wild yeast getting into their yeast stock, which they've bred for their beer production, and contaminating that. And they also worry about organisms getting into their beer and causing contamination. So it's a serious problem. So when you're saying the process is similar to brewing beer, your yeast are out in the open. And then the picture on the right is Sapphire Energy, which is an algae company that actually recently signed a deal with Monsanto. And it's just a computer image graphic of what their 300-acre open pond algae farm would look like. This is being built with help from money from the Department of Energy and a loan guarantee from the Department of Agriculture from the Rural Advancement Program. And when you're working with algae, which can easily become aerosolized in an open pond for up to 300 acres of open pond or 70 square miles, whatever it is, those algae are out in the open. In the slightest breeze, they can get out. If a bird lands on the pond, flies wherever it goes, If you're working at those scales, you're working at the commercial scale, you can pretty much be sure that your organisms are going to escape into the environment. And don't just take it from me. These are quotes from one New York Times article about algae specifically. Two leaders in the algae, I think both for biofuels actually, saying how algae have already been carried out of the lab on skin, hair, being blown out of an air conditioning system. And then the founder of Green Fuel said, of course, algae are going to leak because people make mistakes. So just to say, if we're working at the commercial scale, these things are going to get out, and some of these organisms have already probably gotten out into the environment. So what would the problem be? First would be genetic contamination. This is kind of similar to what we see in agriculture already with genetically engineered crops. Unlike other types of pollution, like an oil spill, which you can clean up, supposedly, hopefully all of it. Once organisms get out into the environment, once their genomes get out into the environment, it's impossible to clean them up. It's impossible to recall them. So once they get out, they're out there for good. And also, even if the organisms die in the environment, if they're weaker than their wild relatives, their DNA will still remain out in the environment. It doesn't just disappear when the organism dies. It stays out there, and it can get picked up as we're going to look. So genetic contamination can happen in a number of different ways through processes of gene transfer. So that could be an algae swapping genes with a relative algae. It could be an organism swapping genes with an unrelated organism. There's an example. They found a sea slug actually picked up DNA from algae and was able to conduct photosynthesis on its own for some time. So it's not just between similar organisms. As mentioned, through transformation, free DNA in the environment can be picked up, and that DNA could come from dead organisms. And then also transduction, where viruses are swapping genes back and forth between organisms. So there's a number of ways that genetic contamination can happen. I tell this slide, suicide genes to the rescue, with a question mark. A number in the synthetic biology community say that we don't have to worry about genetic contamination, that we'll create suicide genes, or we'll create alternative genetic systems to deal with these risks. So if the organisms escape, then they won't cause any harm. They'll die. There's nothing to worry about. The Presidential Commission on Bioethical Issues that JD mentioned, they didn't seem too concerned about the environmental risks. They said, we'll have suicide genes. But the only research they actually quoted around the idea of quote-unquote suicide genes is still a very experimental, in the lab research project that actually hasn't moved beyond the lab yet. So a suicide gene would be something like engineering a dependence on a certain chemical. So if the organism escapes, it couldn't survive in the wild without that chemical. It wouldn't have its food, so it would die. And then there's also other ideas. I think I heard something come out of DARPA, but we don't know what's actually happening, of creating a kill switch. So you can kill organisms somehow, theoretically, I guess. Again, this is all theoretical. We don't know what it will look like, but some folks are saying this is going to solve the problems. And there's also attempts to create alternative genetic systems. So some are working on xenobiology, creating DNA and RNA with different sugar backbones, or engineering different chemicals into the base pairs that aren't found in nature. So if they escape, they wouldn't be able to swap genes with wild organisms. Fortunately, these attempts are going to have a whole slew of their own problems. For example, when we're talking about creating new nucleotide bases and base pairs, some researchers recently, within the past few months, successfully replaced thymine with a chemical known as 5-chloroacyl in the microbe's DNA. The thought is that that chemical is toxic to most organisms, and so other organisms won't pick up that DNA because it would be toxic to them, which makes sense. But, of course, when the organism dies, there's still going to be the 5-chloroacyl in the environment, possibly. So just risks that haven't been looked at yet. And then it's just also important to note, again, that this is all completely theoretical at this point, and nothing has been proven to work at the commercial scale. Another set of concerns would be if these synthetic organisms, become a new class of invasive species. This quote up here, which I'm going to take a second to read, is from Dr. Snow, who was the only ecologist to speak to the President's Commission when they were researching synthetic biology. She said this was her worst-case scenario, that algae grown in open ponds will be engineered to be very hardy, and they could be more competitive than native strains. The new type of engineered algae might spread to natural habitats, to lakes, rivers, and estuaries, where it might flourish and displace other species. And in some cases, this could result in an algal bloom that could suffocate fish and release toxic chemicals into the environment. The general idea is that if these organisms get out, they could die, but they also could find an ecological niche and could possibly take over a certain part of an ecosystem. They could also become new pollutants. Just as completely hypothetical questions, what happens if algae that are engineered to produce oil or petroleum get out into a local waterway or an ocean, and they survive somehow, and they keep spitting out oil into the ocean? Or what if yeast that are engineered to break down biomass, say trees or corn, gets out into a local farm? Again, thankfully none of this has happened. It's all theoretical since the field's still in a pretty early stage. But I think it's still important to ask these questions and to try to think about what those risks might be ahead of time. And then just quickly touch on some of the public health risks. JD had already mentioned DIY bio and iGEM, and thankfully, thanks to Todd and some others, hopefully iGEM's going to have some better safeguards in place. One main concern is around worker safety. Is the workers that are actually in these labs, they're in an open pond algae farm. What happens if they breathe in these organisms? It gets into their eyes or whatever. If they carry it onto their clothes, bring it home to their families or into local communities. As far as we can tell, there haven't been any actual comprehensive risk assessments to look at what some of these risks might be. If you have them, please send them my way, but I haven't been able to find them. There's also biosecurity concerns. As was mentioned, things such as the 1918 Spanish influenza have been recreated in a lab using synthetic biology tools. And there are some concerns, quite serious concerns, enough so that the FBI is involved and is talking to these communities to make sure nothing bad happens. But there is, of course, still the risk, particularly as the technology advances and it spreads more and more to people through things such as do-it-yourself biology, where people can create organisms in their garages. And then also risks around altering the human genome or even the human microbiome. Prominent synthetic biologists such as Jurendi, George Church, Craig Venter have talked about how we could possibly one day re-engineer the human genome for things to fight diseases or even to, quote-unquote, improve the human, make us smarter, faster, stronger, whatever. And then also the idea we can engineer the microbiome, so all the microorganisms that live in our bodies that we are completely dependent on, if we could possibly re-engineer those for fighting disease and other things like that. But again, I think the risks have not been looked at for what the impact these synthetic organisms might have on human health. Of course, most of them will probably be benign, but there's a chance that some of them won't be. And how do we deal with those risks moving forward, especially if we're working at a commercial scale, working with thousands of new organisms that are being created every day? In the U.S., the only synthetic biology specific regulation on the books comes out of HHS, where it's a voluntary guideline for DNA synthesis companies to screen their orders to identify things that might be on the select agents and toxins list. So to try to prevent people from creating things that could be used as a bioweapon or others. But again, that's voluntary. Other agencies are trying to fit synthetic biology into other laws and regulations through the Coordinated Framework for Biotechnology. But a lot of these regulations were written well before people even had the idea of synthetic biology. Laws such as TSCA, the Toxic Substance Control Act, and others where these laws were written really not with the ability to assess the risk ahead of time or to deal with the risk and mitigate them when you're dealing with these new risks from synthetic biology. I would even argue that they're outdated for other old forms of biotechnology too, but we can talk about that later. I got these numbers on government spending from the Wilson Center. You can probably ask Todd more and correct me if I'm misrepresenting them, but they found that the U.S. government spent hundreds of millions of dollars on synthetic biology, I think since 2005 or 2006. 4% of that funding went to research ethical, legal, and societal issues around synthetic biology. And at least publicly, none of that money was spent on environmental risk assessment. That doesn't mean it's not happening, but they're not making it public. It'd be great if they did. And then of that, the Department of Energy is by far the largest funder of synthetic biology in the U.S. government, spending $600 million between 2006 and 2010. And what's interesting is also a couple months ago in the Federal Register, we found that the Department of Energy was trying to get exemption from the National Environmental Policy Act, trying to get exemptions for environmental risk assessments for many of their research projects. And since a lot of synthetic biology work that's being funded by DOE is still still considered research, and the types of projects they were targeting, a lot of that research that's happening right now under DOE funding, if this rule is finalized, would not have to go through an environmental assessment or environmental impact statement. So I think that's incredibly troubling, particularly as we move forward and the technology grows. And then lastly, probably the more, I guess, controversial part is what Friends of the Earth would recommend. And I think the groups here that are talking would agree with these. There is still contention about what we can actually do about this. And so we, at Friends of the Earth, we try to support a strong application of the precautionary principle. And so to us, that would mean that there should be a moratorium on the environmental release and commercial use of synthetic organisms until these 3 points are met. So that would be proper risk assessments conducted by governments with full participation of the public and open participation, until the health, environmental, and socioeconomic risks have been identified and mitigated, and until we actually have laws in place to deal with these risks, and laws that are able to evolve and that are flexible enough to deal with new advances in the technology. This doesn't mean that we think all synthetic biology work should stop. We think it could be useful for helping us to better understand biological systems and how they work. But right now, we think the risk is too large for the research to move beyond the lab. So if you can contain your synthetic organisms and you can ensure that it doesn't harm your workers and doesn't harm the public or the environment, go ahead, but just make sure it stops right there at the laboratory door. And with that, I will pass it off to Kathy Jo to talk about some of the more broader socioeconomic issues. Thanks so much.
So the speed of my presentation will be somewhere between JD and Eric's speed. For those of you who may not know, at Etc. Group, we're a small international civil society organization based in Ottawa, in Canada, and we're often described as a technology watchdog, but we never look at technologies without also considering the issues of ownership and control. That can mean who's got the patents, or who has the biggest market share, or who has control over raw materials. And that's the piece that I'm going to talk about today, the corporate sectors involved in synthetic biology and what the implications are of that involvement. But before I do that, I'll explain the lens through which we see the development of synthetic biology. This description from Aristides Patrinos, who's the president of Synthetic Genomics, is more than 5 years old now, but it's still one of the clearest expressions of the synthetic biology vision, which is to produce a high-value product, in this case, transport fuel, using an easy-to-come-by feedstock, such as a hardy perennial grass. And then what happens in the vat is a transformation from plant to fuel, and that is done by synthetic microbes. When we first tried to think about how this could work and what that magical vat might look like, we could only imagine a machine that might have come from a Dr. Seuss book, something comparable to Sylvester McMonkey McBean's machine that can put stars on or take stars off the bellies of Sneetches, depending on what's the fashion of the day. And we worked with an artist activist who's in the UK, and he created this cartoon, which is fantastical, but it does include all the elements of the vision, and then some elements of our more dystopic vision. The synthetic biologist is up there in his lab programming microbial genetics and tweaking the organisms according to what kind of feedstock needs converting into what kind of product. And those engineered microbes that do the work are often referred to as living chemical factories, and they will be patented. There's the vat that's holding the proprietary microbes, and they're doing their work. And the way we've shown it here, the vat is providing less than perfect containment. They're the final products: plastics, fuels, pharmaceuticals. And like Patrinos imagined, the fuel goes right into the gas tank from the vat without having to make modifications to the fuel or to the car engine. There's the CEO, of course, who's turning the profit. And there's the feedstock, in this case, a bit of tree. But there are some crops nearby. And here we're showing a farm family getting sucked up with the feedstock. And that points to one of our biggest concerns about synthetic biology and the lens through which we view its potential socio-economic implications and environmental implications, which is where will the biomass feedstocks come from and who will control them? Plant-based feedstocks for synthetic biology manufacturing platforms are usually referred to as biomass. That's the green stuff that was once living or is still living. The term narrowly refers to the weight of living matter, plants, animals, bacteria, fungi, found in a specific environment. But now biomass is more commonly used as a general term to refer to the use of non-fossilized biological and waste materials as a feedstock for the production of fuels, chemicals, heat, and power. So biomass is a general term, but it's also a measurement, and it implies a particular way of thinking about nature as commodified even before it enters the commercial market. So everything living is a potential article of commerce. In terms of corporate control, then, we're really asking who will become the new biomasters, and what kind of biomass, and where will it come from? And that question, where will it come from, is a real question, but it's also somewhat rhetorical because we know that most of the Earth's terrestrial biomass is found in the global south. The map here on top is a product of the Wilson Center Synthetic Biology Project, and it shows the concentration of synthetic biology labs and research in the U.S. and Europe. And the bottom map is from the Oak Ridge National Lab, and it shows the areas of the planet where living biomass is most dense, and you can see that it's in the global south. You can also see it in a different view where you can see where the green stuff is found. The global south accounts for 68% of the world's forest biomass, and using trees to produce energy dates back hundreds of thousands of years. But the hope is that synthetic biology, perhaps in conjunction with genetically modified trees that are engineered to have low lignin content or to be super fast growing, will produce microbes that can help break down the cellulose in trees to make them a more efficient energy source. And there are the so-called energy crops, cane sugar or jatropha or oil palm or corn. And the so-called crop residues are another source envisioned for biomass feedstocks. Last month, the Department of Energy published an update of its 2005 study on biomass feedstock availability. And both studies, the 2005 study and the updated study, evaluated the potential crop and woody biomass availability in the U.S. through 2030. The initial study tried to determine whether the land resources in the U.S. are capable of producing a sustainable supply of biomass to displace 30% or more of the country's petroleum consumption by 2030. And it concluded that it was possible to produce 1.3 billion tons per year. Hence, it's referred to as the billion-ton study, mostly from agriculture. In the updated study, they revised their conclusions. It still concludes that there's enough biomass, but that the first study had overestimated how much feedstock could be gotten from crop and forest residue, but had underestimated the energy crop potential because of projected higher planted acreage. But we've already seen the global consequences of expanding cropland for the first generation of energy crops. Most assessments, including by the World Bank and the Food and Agriculture Organization, have concluded that biofuels were a significant factor in the sharp rise in food prices in 2008 and in the current rise. In 2004, biofuels consumed just 2% of world grain and virtually no vegetable oil. But last year, biofuels consumed more than 6.5% of grain and 8% of vegetable oil. Governments worldwide want to triple production of biofuels by 2020, And according to a Princeton researcher who's looked at this, he says that means moderately high prices after good growing years and soaring prices after bad ones. So the question is a simple one. Do we have enough biomass? It's estimated that the Earth produces 230 billion tons of biomass annually. We already use an estimated 1/4 of the Earth's biomass, and ecosystems are stretched to the limit, and in some cases beyond the limit. It happens that yesterday, September 27th, was designated as Earth Overshoot Day, according to the New Economics Foundation, which calculates the day during the year on which the total resources consumed by humanity will exceed the capacity for the Earth to generate those resources that year. And somehow we're doing better than last year when Earth Overshoot Day was in August. But still, the point that they are trying to make is that we are already in ecological debt. We've already used up more than than the Earth can renew. Growing crops for energy production, in our view, won't solve the problem of peak oil or climate change. This is an industrial-scale biorefinery in Tennessee that uses synthetically altered yeast to ferment corn sugars to produce a bioplastic for DuPont. DuPont says that the biofiber will eventually replace nylon and requires 40% less petroleum to produce. However, this one factory uses 40,000 acres of corn. That's 6.4 million bushels to produce 100 million pounds of the plastic. And I haven't been able to find out how quickly the refinery goes through those 6.4 million bushels of corn. But BIO, the U.S.-based biotechnology industry organization, estimates that what they call a moderately sized biofuel refinery producing 65 million gallons per year needs at least 500,000 acres of cropland to sustain it for a year. So it seems like that's about the size of Connecticut for one plant. These concerns, the high food prices, strained ecosystems, are part of the reason that algae is getting a lot of media attention and funding right now. And the rationale is that algae are fast-growing, they're more renewable, they're relatively easy to engineer, and they aren't directly implicated in the food versus fuel conundrum. And there's hope that synthetic biology will make it economical to make biofuels from algae. And since 2000, more than $2 billion in private money has been invested in the field in the U.S. But the algae that will produce biofuels won't be harvested like this. It will look more like this. Building these shallow ponds, JD mentioned how big they might have to be, or greenhouses, filling them with synthetic algae strains that can produce higher amounts of oil than natural strains. Algae aren't exempt from land use and resource use concerns, particularly water, but the biggest, in our view, the immediate concerns with synthetic algae are the environmental contamination concerns that Eric raised earlier. So the idea is that we're going to be shifting from a petroleum-based economy to a bio-based economy, and that the shift is already underway. But we need some perspective, I think, on how far the pendulum has swung. Swung? Swing? Swang? Swung? In 2010, the world's energy consumption grew by 5.6%, faster than any year since 1973. Fossil fuels accounted for 88% of the world's primary energy. So-called renewables, including nuclear power, accounted for the remaining 12%. Even though world biofuels production grew by 14% in 2010, it accounted for just 1/2 of 1% of global primary energy consumption. So 0.5%. And we're already, seeing the effects on land and food prices from this use of biofuels that contribute 0.5%. The top 10 energy companies, the biggest 10 energy companies in the world, account for about 25% of about $7 trillion energy market. And a lot of these companies, like we've already heard from JD, are high-profile investors in synthetic biology. And yes, they want a cleaner, greener image, but it's also true that peak oil is a reality, and extraction is getting more and more challenging. So I'm not saying that that it's all a charade. I think there really are serious investments in this, but petroleum is still king by a long shot. But the companies are aware that future profits will depend on diversifying and controlling bio-based feedstocks for energy production. And it's not just about energy. A company like Amyris, which JD talked about, has deals with big oil, but also big agribusiness. Those companies there on the lower right, all of those are involved with sugarcane, which is no surprise since that's the feedstock that Amyris is using. They have deals with Procter & Gamble and with this Gruppo M&G is an Italian company and one of the world's largest producers of PET plastics. And another company, Solazyme, is another SynBio startup, and it's not limiting its focus to energy either. The company says its market areas are fuels, chemicals, nutrition, and health sciences. And it has deals with Dow Chemical to produce a chemical lubricant, Unilever to develop oil for soaps and other personal care products, Chevron and the U.S. Navy and Department of Defense, and Bunge, a grain sugar and sugar cane processor. In March, as
Solazyme just launched a new IPO, in fact.
In March, Monsanto announced a multi-year collaboration with Sapphire Energy, as Eric mentioned, and Sapphire is also working with algae. The amount of the investment wasn't disclosed, but Monsanto now has an equity stake in the company. The news releases that announced the deal were revealing because Monsanto was quite clear that they weren't interested in algae, but they are interested in corn, cotton, and soybeans, and they're hoping that Sapphire's genetic tweaks of algae will be applicable to the commercial crops that they already monopolize. And Sapphire's CEO was clear that his company wasn't only interested in energy, that they're not just focused on producing one type of a high-value compound, but or changing one thing into another, as he says, but about creating a whole new commercial agriculture. And if there's anyone who knows anything about creating a new commercial agriculture, it's Monsanto. I just want to remind you that Monsanto is the world's largest seed company and the world's fourth largest pesticide company. And it was a new technology, the technology of genetic engineering, that allowed that to happen because they could then sell their seeds and their chemicals as a package deal. When they genetically modified seed to survive a dousing of Roundup, they were then able to monopolize, and they do, genetically modified soybeans first. And so the question is, will synthetic biology help Monsanto to create a whole new commercial agriculture, and what will that look like? And then if that happens, maybe we shouldn't be talking about new biomasters at all, but old biomasters, and the old biomasters with a new technology that helps them hold on to and increase market share. Thank you.
Well, thanks, Kathy Jo. I'm going to turn it over to Dave Rajewski now, who's going to give you a brief.
Switch computers.
Yeah. Oh, does anyone have a burning question or two, or that we can answer quickly while we switch out a computer?
What was Solazyme's IPO? I didn't know about that.
They basically are looking to get more private investment for the work that they're doing to make both fuels and chemicals.
Restart your computer. I know that's that sometimes.
One all the way in the back.
Hi, my name's Amanda Wilson with Interpress Service, and last summer in Europe there was an E. coli contamination that was traced to an organic farm, and they found out that it came from sprouts, and there was mass hysteria in Europe. I was actually there, and I just wondered if you had any ideas about, whether that's related to what you've been talking about at all, or I mean, maybe how to talk about these issues without seeming like it's a conspiracy theory. Because when that came to mind, I just thought it's crazy. It couldn't be true. But I mean, do you have any idea about that case?
Well, I think in my day job at this, I also work on food pathogens. I think the challenge of all of the food pathogens is a good reminder. These are natural organisms that evolve rather quickly, and they've evolved quickly, in my opinion, as part of our increasingly industrialized agricultural system. We had in the U.S. one of the largest recalls of ground turkey, 36 million pounds of ground turkey with a strain of salmonella that has become antibiotic resistant because of our feeding antibiotics to all these animals that we raise. These strains, whether they're E. coli or Listeria or Salmonella, are evolving faster than our ability to control them. And so one of the challenges I think that we've got with synthetic organisms is to understand this aspect of these organisms, that they can evolve rather quickly, and make sure that we don't accidentally, nobody, I believe, went out and developed intentionally the strains of E. coli that can kill people so quickly. But our agricultural system has, accidentally. And as we develop a new system based on, in some cases, E. coli or Salmonella are the working parts of this new system because they reproduce well. We need to make sure that we don't accidentally wake up one day and find them in a vat of beans.
All right, I'm going to just sort of buzz through this pretty quickly. Every year we do focus groups, and then we also do national surveys about what do people on the street think about synthetic biology. And these we've been doing for years and years now, also with nanotech. So we just did focus groups up in Baltimore, and I'm going to just go quickly through some of the highlights. focus groups are a sample of America. So this one up there had a chef, a patent examiner, a bunch of stay-at-home moms, a flooring contractor. So the idea is not to take a huge sample, but one that's kind of representative of the American population, high schools, graduates to university, to grad schools. Religious convictions were 40% very strong, 30% not so strong. 2 hours each focus group, so we go through a very elaborate script with these people. Only one person out of all the people in the focus groups had ever even heard of synthetic biology when we started. So, pardon? There were 8 people per group. So when we were done, probably they knew more than 99% of the American population, because they did get a lot of data. Focus groups, interactive, they're nonverbal. Cues, you can be useful. They're not a statistical sample, so there's a lot of caveats here. They are very good for exploring new stuff because you can kind of drill down and follow people's discussion threads. Small sample size. It's obviously peer pressure in the group. Again, we spend lots of times, a month or so, developing the script. The people that do this are trained to do it very well. What is it? Okay, here's what people thought it was. No information. Man-made materials, genetically modified plants. The one thing that comes up again and again, which surprised me, was they keep talking about regenerative medicine, prosthetics devices, man-made limbs. This was completely unsolicited, right? So they're making some kind of association with medical applications. Biological parts, we're going to replace joints. These weren't a bunch of old folks waiting to be kind of regenerated, but there was a bunch of kids in here, but this was quite interesting because it wasn't the usual set of associations. What are the initial reactions? They're very schizophrenic about this. I'm excited but freaked out. It's weird, but it's creative. It tends to be very case-specific when they talk about it. Medical field gets a lot of interest. They don't like the idea of just using it for cosmetics, just to kind of soup something up. Don't like the idea of making food grow in places it doesn't belong. Some people just have a real hard problem about creating anything whose parent is a computer, so you run into some real ethical pushbacks. Concerns, kind of once they started to learn about this. Mutation, self-replication, people were worried about this issue of a kill switch. They asked some valid questions about, okay, prove to me that you can actually make this work. That there is such a thing as a kill switch, that you can shut these organisms off. Who's going to regulate it? Don't think the ramifications will be short-term. We've heard that before. Some people just didn't like it, period. This was just from the national poll we did last year, just to give you a sense of kind of where people come out about what they worry about. Biosecurity is up there, ethics and biosafety. So this is, again, a statistically valid poll. Messages, Matt, we actually pulled messages directly from the literature, from public pronouncements. We took the people off and tested them with the public here. The huge differences in the responses to messages. These are just 2 messages about synthetic biology. This one was seen as very positive. This one was seen as very negative. This whole idea of tying it to intelligent design. So don't assume any message that you have is going to work out. I mean, we were quite surprised by both the benefit messages and the risk messages, what actually resonated with people. Assumptions we made were mostly wrong. Metaphors matter. Some of you realize that people are coming at this from different disciplines, so they talk to each other using analogies. So you've got a whole bunch of analogies coming out of the computer science area. So we tested these metaphors. The ones that come out neutral or positive tend to be software, computing, machine circuits. The ones that come out negative, factories, biobricks, living foundry, Legos. Part of the problem with these is sometimes they're so confusing, nobody knows what a living foundry is. So as soon as people start to think about that, sort of doubt creeps into their head, and they start to go negative on you. So if people can make a fairly fast association, and software seems to be clean, fast, innovative, modern, that gets high points, right? If they're talking about factories pollute, foundries pollute, they were dirty, why are we talking about that? The more they had to think about the metaphors, the worse they got in terms of associations. Base organisms mattered. There's a huge difference if you're building off of yeast. We put it on our bread, we eat it, we drink it, yeast is good. Right? These are direct quotes from them. You start building things off of E. coli, you've got a different problem altogether. More scary, you don't have it in your cabinet necessarily. You hope you don't. Probably do. But again, there's just when people hear about E. coli, quite often it's in association with food poisoning and nasty things. So what we choose to build things off, I'm not saying the yeast-based stuff is perfect, but as soon as you choose an organism, you're choosing a set of associations. You might be better choosing an organism that's known by no one than to choose one like this. I'm okay if they take the dangerous thing out of it, if they can do that. We tested on, what do you think we should use this for? These are the top ones. Again, this is not a statistically valid sample, but it reflects a lot of what we saw in national polls. Cancer cures, cleaning the environment, biofuels, malaria drugs, sensing harmful contaminants. Who should provide oversight? Here's the votes. Amazing that Congress was even mentioned here. Somehow they seem to trust scientific bodies. The federal government still ranks high. For those of you who are here from the government, university scientists, NGOs, less trust in the companies that are actually developing the applications, and of course the Congress. We didn't check with the president, but last time we did it came down low. So takeaway lessons. Again, we have a lot of data here. We've got 4 hours of video transcripts we're going through, and we can share a lot more of this with you. But they don't know much about it. You can start from that assumption. If they don't know much about it, what if scenarios, ethical considerations, a lot of that just begins to fuel negative feelings. Applications to medicine and environment seem to be more compelling. Emphasis of benefits to people's lives, enhanced support, messages that play on fears and unintended consequences stoke opposition. Using positive tone and comprehensive explanations, layman's terms is actually important over scientific jargon. The regulation, doing things in contained environments, if you can do that, oversight were seen as keys to actually reducing fears about unintended consequences. Here was one message that we ended up at the end with. So again, I just wanted to give you kind of a quick overview, but we have a lot more data we're mining right now. So you want to just do questions?
Does anyone have any questions you can address to anyone? Just wait for a microphone.
Thank you, Daryl Ditz. I want to say I'm feeling this thing in the pit of my stomach, which maybe is I'm a little freaked out, using the language of the focus group. And I remember this feeling in this building before on Nano, where I feel like I find out about something I should have worried about long in the past, and now it's too late. I wonder if any or all of you might want to comment a little bit on the practical prospects for some kind of control. Industry guidelines, we've got some of those out there. Regulation, hard to be too optimistic about that. International coordination around this. Is there anything you can say to make my stomach feel better?
One thing that I'll say on the international front is last year at the UN Convention on Biological Diversity, the convention looked at synthetic biology, and that was the first time an international body looked at it outside of the Bioweapons Convention, I believe. And they'll be re-examining this coming April at their scientific and technical review body, and then probably again at the next COP. So that'll be the next conference of the parties, I believe, October 2012. So there, at the international level, there are conversations that are starting about what are the impacts of synthetic biology on biodiversity, but also human well-being, and that includes a lot of the socio-economic issues that Kathy Jo raised before. In terms of actual U.S. regulation or oversight, I'm a little less optimistic. The President's Commission on Bioethical Issues is a very similar quote to that. The continue to go forward, but please be careful. They coined this term prudent vigilance to replace the precautionary principle, or they said the opposite of that was the proactionary principle or something. As far as we can tell, the only time that term prudent vigilance was used before was when the chairwoman's husband used it in peer-reviewed literature. And so they're trying to replace principles that have been established in international law for a number of decades and even U.S. law for decades. So I'm a little less sure what will happen. There is a new synthetic biology caucus in the House of Representatives, so we'll see where that goes and what comes out of that.
And if you haven't heard of the Convention on Biological Diversity, that that, I mean, you shouldn't feel bad because although
the U.S. is one of the few nations that's not a signatory to it, so those of you that lobby Congress, you might remind your members of Congress that it would be nice if the U.S. were a signatory. It goes and lobbies the other members, but it would be nice if the U.S. actually were bound by what they decide.
Yeah, I think the U.S., the Vatican, and Andorra are the only UN nations not to, or the UN parties not to have signed it, or not to have ratified it. I think we signed it, but we didn't ratify it.
There was a question up front.
Yeah, Colleen had a question. Across you.
About a decade ago or 2 decades ago, there was a bit of back and forth about the kind of intellectual property protections that genetically modified organisms get under current IP laws. Has anybody thought through about the extent to which this synthetic biology would make it very easy to get around existing IP? Because you have all this, junk DNA. Insofar as existing IP has a particular structure rather than a particular function, it will allow you basically to modify useless structure and then say it's different and then kill your existing IP. I mean, when you do genetics and things like that, you're always looking for small variations, and the extent to which you can do, your knowledge is good enough to do the synthetic biology. Wouldn't that make it very easy to get around existing IP?
Well, I think you should actually go to the Patent Office database, and you'll find that all of the researchers that I've mentioned are patenting their work. They're patenting what they call their both the process and processes.
I want to do new things, but this is somebody who wants to get around Monsanto's stuff right now and use that. So
well, what's going to get around Monsanto stuff is herbicide resistance as they're moving, as weeds. It turns out they evolve. Maybe Monsanto and the religious right are the two groups that don't believe in evolution. Mm. But it turns out these weeds, when sprayed with the same chemicals over and over again, develop resistance. There is a case now going through the courts on the patenting of genes that correlate with two genes for breast cancer, and that will likely go to the Supreme Court next year. If the court rules as we wish it would, and it's hard to know with this court, if they rule that naturally occurring DNA is not patentable, some of the efforts by some of the researchers to collect lots of DNA that they can patent and use in their synthetic organisms would be a little thwarted. I don't know that really doesn't really answer your question. Well, both. I mean, the questions the court's going to have to deal with is DNA itself patentable? Is cDNA patentable? And they'll probably also later get a case, is synthetic printed DNA patentable if you're using the same sequences as found in nature?
My organization hasn't looked at the issue in the way that you described it, but we are kind of looking at it from the other end. And that's one of the things that the Convention on Biological Diversity does is has been working on for decades, is this an agreement for access and benefit sharing. So this is an attempt to protect groups indigenous groups who have been preserving biodiversity, so that when a pharmaceutical company comes and takes that biodiversity, that there's some kind of reciprocity, that they get something from it. And so one of our concerns is that synthetic biology is a way. We don't believe that access and benefit sharing agreements are very beneficial for these communities. So, but it can provide a way to circumvent those agreements, because, you know, you go to the field, you pluck it, you get its sequence, you tweak it digitally, and then you say that you've got a different organism and that you don't need to give anything for this thing that you got. So that's a slightly different way of looking at it, but it's I think the same issue that you're getting at.
I'll comment on that a little bit. That's actually sort of a debate going on within the synthetic biology community now, is that there's sort of two camps. There's sort of the patent camp that kind of wants to patent every little piece and every little part and every little combination. And then there's another sort of side to that where it's coming from an open access standpoint. There's been arguments about if you start patenting every little piece of this, you're going to almost sort of shut down the whole technology if you need to, you know, get your way through 1,000 different patent thickets in order to combine things. So part of what the iGEM competition is based off of is that the teams are required to submit their parts into that registry, which are then free and open access for anyone to use. So that's one model, sort of from an open, you can sort of think of an open access sort of freeware from a computer software type of thing. But it's a debate that's happening now, and whichever way it falls out, I think will have huge implications on how the technology progresses forward. There's also the issue of whether the patent office can handle this type of thing as well.
There are a couple of places that it might. One is it makes it easier to re-examine patents. Our organization, under the existing patent law, has challenged, a few patents. We got a university in Texas to withdraw its patent on a beagle. The only inventive step that it made was it infected the beagle with an organism and said it was a new organism. The patent office itself withdrew a patent on an infected rabbit that we also challenged. So if it's the new procedures will make it easier for both companies and groups like ours to challenge patents for being obvious or not patentable.
I think we have time for one more question. I think there was one right up front here.
Along the lines of messages matter, I'm not quite sure what the focus groups, what the point of them, what the purpose is for the focus groups. But my question is really around synthetic biology as the term that's used, because I would wonder if there's anyone in the room here, let alone on the panel, who would disagree that it would be more accurately termed synthetic biotechnology. And as a former reporter, I wrote a lot of stories about scientists just feeling that, and the country at large feeling like science is a whole lot harder to say we should stop doing science than we should stop a particular technology. So I think that the message there is very strong. Synthetic biology is the study of something. It's biology. But what everything we've been, you've presented here is synthetic biotechnology. So is it possible for people to, if you would agree with that, why don't we all start calling it synthetic biotechnology?
I am really grateful to the synthetic biotechnologist for calling this synthetic biology, because of the reaction that so many of the public have that wait a second, synthetic biology, this is an oxymoron. I'm not presenting this as synthetic biology is my term of art. I'm calling it synthetic biology because the practitioners are calling it that. And I'm grateful because it gives me a chance to raise questions about it with the public that if they called it metabolic engineering, the public's eyes would just glaze over.
But if everybody called it synthetic biotechnology, I think the public would indeed immediately have some questions and some reactions. And since it's a more accurate term, it just seems deceptive to call it synthetic biology.
And I will add that, at least from the conversations I've had with those in the synthetic biology community, they still debate what they should call themselves, and a lot of them hate the term synthetic biology as well and wish that they had named themselves something different. But also, I think of interest is when talking to funders, a lot of the synthetic biology companies will say how what they're doing is so brand new, it's never been done before. But then when they talk to regulators, they say, oh no, this is just old biotechnology. It's just like genetic engineering. It's the same thing, no need for new regulations. And so they're trying to play both ways. But I do think it's a very valid point that we should also be careful how we talk about it, keeping in mind how the public will receive it, for sure.
Yeah, I don't think the public response to the words are very, very different from the ones we got with nanotechnology. So when we use nanotechnology in focus groups, people would sort of say, small technology, and sort of tell us about it. Synthetic biology hits every third rail that you can possibly imagine very quickly because people start to parse synthetic biology to artificial life. Is that cloning? Is it stem cells, GMOs? And I think the scientific community knows that. I mean, the term was first used by two Polish scientists in an article in the late 50s, I think, and they've kind of inherited it. But I think it's very difficult to get rid of it now. But I think the public, I mean, the reactions when we do the focus groups now versus the ones we've done for years on nanotechnology. Nanotechnology was starting with kind of a clean slate. It wasn't, you sort of had to define it. This one, you start with an incredible amount of negative loading just based on these two words coming together, synthetic and biology. It's not kind of oxymoronic at some level. And I agree, most of it is technology, but I don't know how essentially you change the term right now because I think a lot of the scientists would like to, but they're kind of wedded to it. And also to the extent you keep I would say, combining more and more funding opportunities to the term kind of causes a kind of lock-in effect, the same way we had with nanotechnology. An awful lot of people who were doing microtechnology became nanotechnologies very, very quickly, as soon as millions or hundreds of millions of dollars kind of flowed in there. Now I do nanotech. So I think it's almost a self-perpetuating thing. But I would agree a lot of what you're seeing is technology.
Yeah, I think it's a really good point. We started out calling it extreme genetic engineering, and that didn't really catch on. But in fact, I think that's what it is. I was at a meeting with people who are doing this kind of work, and some of the people who were also doing genetic engineering, and there was kind of a little bit of a heated debate because a lot of people in the room were calling it genetic engineering plus, and one synthetic biologist just stood up and he couldn't stand it anymore, and he was saying, stop calling it that, we're doing something new. And so in a way, I think genetic engineering plus is also more accurate. I think synthetic biotechnology is more accurate. But I think all the things that Dave said about funding and it would be hard. But that doesn't mean that we can't call it synthetic biotechnology or extreme genetic engineering.
Regardless, we want to leverage the conversation to do more real oversight of it when the President's Commission on bioethical issues says that the coordinated framework will work to take care of this. They're making an amazing leap. They're assuming the coordinated framework works to take care of the old kind of genetic engineering, not to say anything about it. It doesn't work to take care of the old kind, and hopefully, whatever we call it, we can use it to leverage to get some better oversight than of this new and I think more powerful technology potentially that does need more oversight.
All right, well, we reached the 2 o'clock hour. I want to thank you all for coming, and I want to thank our panelists for talking with us today.